So today the podcast is going to be on a topic near and dear to me and my laboratory over the years, and that is the effects of intermittent fasting on the brain. And my guest is a former postdoc and now a full professor at Latrobe University in Melbourne, Australia, Thiruma Arumugam, or Gary, as we call him. He's a professor of physiology and pharmacology there, and he has an interesting kind of history, unlike myself, who's born and raised in the United States and never left as far as living. He's kind of moved around the world. So Gary, welcome.
Thank you so much, Dr. Madsen. First of all, I'm delighted to participate in this podcast conversation with someone who has not only made significant, contribution to the field of neuroscience, but has also been my mentor for many years. Thank you, Dr. Madsen.
Yeah. And so you were born in Sri Lanka?
Yes, I was born in Sri Lanka, eastern part of Sri Lanka. It's a small town called Batticaloa. And my parents were teachers. My dad was a school principal, and my mom was a maths teacher for primary school students. And so they were interested, all of us, doing well in studies and achieving high. So that was my background. As you know, that Sri Lanka was going through civil war at the time when I was growing up. So it was quite a tough time for us to, like other part of the world where students focus on studies all the time and do well, and we were running around and hiding and all these things. Somehow I got into med school in early 90s, 1990. And after my med school, I wanted to start and finish my med school, but I couldn't stay there because of the civil war. So I left Sri Lanka to Singapore first. And from Singapore to Australia as a refugee, that was in 1992. So it was a quite tough journey for the 2 years, that period. And then I couldn't start going to school straight away because of the paperwork. As you know, when you arrive as a refugee in another country, that you have to go through paperwork. And it took 2.5 years before I got my permanent residency in Australia. And then I started going to the University of Sydney and did Bachelor of Medical Science, and then PhD in pharmacology at the University of Queensland, and another state in Australia. Of course, from there, I moved to Louisiana because my PhD was focusing on ischemia and reperfusion, the model, and studying the effect of complement 5A receptor antagonist. So I wanted to go to the lab where we learn more about ischemia reperfusion injury. So I went to Neil
and at Queensland, yeah. You were, if I look back at your public, you were working on ischemia of the intestines, is that right? And inflammation?
Yeah, I was looking into the effect of complement 5A receptor antagonist in intestinal ischemia reperfusion model, and kidney, liver, so 3 organs. And I published a number of good papers because that drug was very novel at the time. First drug ever to have such a high effect against complement 5A receptor.
You want to briefly, kind of, for people who don't have immunology background, what is complement and why and how might it contribute to damage to tissue when the blood supply is reduced?
Yeah, so as all know that, we have innate immune system, which is really helpful when you suffer from infection. So this is part of our innate immune system. And when complement is a protein cascade activated that produce number of factors, complement factors, that is important when you suffer from infection or infection from foreign organisms such as bacteria and virus, etc. But overactivation of this complement pathway can also damage, harmful to our cells. And that's what happened under disease condition. Activation of complement system produce complement 5A and also, the MAC complex, which is membrane attack complex, that can cause damage to our cells and organs. And that's what you see normally in disease conditions such as ischemia, reperfusion, stroke, even dementia. So controlling complement activation, it's important. And so the drug to target most potent complement factor, which is C5a, its activation. So we produced that drug, and I was testing the effect of that drug against the different ischemia reperfusion model.
Yeah.
So at the time, I was looking at who is expert of ischemia reperfusion model. I found Neil Granger in Louisiana. So I emailed him, and he accepted, and I started postdoctoral position with him. I was there for almost 2 years. Then I wanted to get into neuroscience, and I was looking around, and I found you, Dr. Madsen, and I called you, and you offered me to come there for an interview. So I arrived in Baltimore. I remember that day. And we had one-to-one interview in your room. And then I had the feeling that you offered me straight away. So that's when I started. It was in 2004, I think August, if I can remember. Yeah.
Yeah. And so that I'll take over for a few minutes here if the listeners can indulge me. Beginning in the mid-1990s, we were trying to understand what goes wrong in the brain in age-related neurodegenerative disorders like Alzheimer's, Parkinson's, also stroke somewhat. And we know that the major risk factor for those are actually the three main killers in terms of brain disorders. Advancing age is the major risk factor. And we had some experimental models back then using neurotoxins and also stroke models similar to what you used when you came to my lab, where you transiently occlude a blood vessel in the brain, kind of mimicking a clot forming and then resolving. And then other labs had shown that caloric restriction or every other day food deprivation, every other day fasting could extend lifespan a lot in rats and mice, in some cases by 50% or more, when it started when the animals are young. So, just kind of simple-minded thinking, okay, if we have some intervention that we know counteracts aging and extends longevity, will that have beneficial effects in our animals, rat and mouse models of Alzheimer's, Parkinson's, and stroke? So, I recruited 3 different postdocs, those projects. So one, actually, Anna Bruce Keller was the first. We had a paper in Annals of Neurology in the late 1990s where she showed in model of epileptic seizures, where she gives them a toxin. Actually, activates glutamate receptors excessively, so it overexcites neurons, and it can cause seizures and even death of neurons. And Anna found if she maintained rats on every other day fasting, and she had to do it for at least a month before she saw a clear effect. And the effect she saw was a clear, highly significant reduction in the damage to neurons in a brain region called the hippocampus, and much less of a deficit in learning and memory associated with the degeneration of those neurons. And then another postdoc, Wenjian Duan, who's now a professor at Johns Hopkins, she used a neurotoxin model of Parkinson's disease. There's a mitochondrial poison called MPTP. It has a very interesting history. If people are interested, they can read my new book on glutamate, Sculptor and Destroyer. But anyway, this toxin will be selectively concentrated in dopaminergic neurons, the neurons that deploy glutamate as their neurotransmitter. And it will cause the mitochondria not to work very well. And that leads to an energy failure in those dopaminergic neurons. They degenerate, and the rats or mice that are given the toxin have problems, just like with Parkinson's disease. They can't control their body movements. They have tremors. And Wenjen found that when she did intermittent fasting before she gave the toxin, she saw less damage to the dopaminergic neurons and improved functional outcome. And then finally, I had another postdoc, Zhaifang Yu, who could do the middle cerebral artery occlusion reperfusion, a model of stroke.
Yeah.
And again, she found beneficial effects. And then we moved. That was one. I was at University of Kentucky. Then I got recruited to National Institute on Aging to start and head a laboratory in neurosciences. And so you came somewhat, 4 years after
you started,
I'd been there. And I wanted to continue work on this intermittent fasting story, trying to get at what are the cellular molecular mechanisms that account for these actually quite remarkable neuroprotective effects. So Gary, then you came to the lab. You want to talk about your project?
Yeah, absolutely. Yeah. So one thing, Dr. Matson, that you mentioned about the existing literature about the effect of calorie restriction and intermittent fasting and lifespan extension, right? That quite interesting. When I was going through the literature, I found the first paper was published in 1935 by Clive Mackay in Journal of Nutrition, where he had this rat model where he put them under calorie restriction, extension of lifespan, and in male and female, it was very significant in male compared to ad libitum, and some extension in female. The female lived longer than male in his studies. And one thing I noticed, like when you look into the key discoveries of aging, they mention everything, but they missed in the paper I was talking about, published in Nature as a review paper, key discoveries in the field of aging. Intermittent fasting, similar to Clive Mackay's work, 1946, there's a paper where every other day intermittent fasting extends the lifespan. So I'm pretty sure you knew about that paper, right? 1946 paper.
Yeah.
Yeah.
And then a little bit closer to the 1990s was Don Ingram's work at the National Institute on Aging, where he did both mice and rats every other day feeding. Yeah.
And I think that before Don Ingram, Richard Wendrich, right? He did a lot of work.
But that was caloric restriction.
Caloric restriction, not intermittent.
It wasn't intermittent fasting. That was fasting. Caloric restriction. Yeah. And actually, it turns out though that in a sense, and this is something the listeners may be very interested in. So the way, quote, calorie restriction, unquote, studies are done is you take like 20 animals, for example, and they're an individual cage, one animal per cage, and you give them food and you essentially weigh the food each day and see how much food each animal eats each day.
Yeah.
Then what you do after you've established how much food each animal is eating, you divide them into two groups. In one group, you give about 30 to 40% less food than they normally would get.
Yeah.
However, in all these calorie restriction studies, Weinrich, all of them, the way they do it is they give the animals their daily food allotment all at once.
Yeah.
And when they're have that big of a reduction in calorie intake, and you give it to them all at once, they eat it all within 4 hours or so.
Yeah.
4 to 6 hours. So they're actually fasting for up to 20 hours every day. And that's now called time restricted eating or time restricted feeding in animal studies. But actually all the previous studies on calorie restriction were also time restricted eating.
Time restricted feeding, yeah.
Yeah.
I agree. And that I think the recent study published in Science by Joseph Takasashi
Yeah, and there was
so
one before that too that clearly documented that.
that's right, yeah.
Yeah.
That came from East Coast of USA. I remember that paper, yeah.
Yeah.
So that's quite interesting. And when I joined your lab, I completely, not aware of the effect of intermittent fasting against any neurological conditions. And I remember that I was sitting in the office with Ruchin Van, and Ruchin came up with this idea of testing the effect of every other day fasting against stroke model. And as you know, I still put my picture and your picture in early 2004 in my talk to show how, I was food positive, and you were practicing intermittent fasting or calorie restriction at the time. And I wasn't big fan of, intermittent fasting or calorie restriction. But I listened, you and Ruchin started testing the effect of every other day fasting against stroke. And quite interesting in this study, what we have done, three different groups: young animals, middle-aged, and old animal. And we put them under everyday intermittent fasting for around 6 months, up to, we wanted to do it 4 months, but by the time we started doing stroke in these animals, there are quite a lot of number of animals. I remember over 200 or 250 animals, if I can remember correctly. And so we induced stroke, and what we found that intermittent fast animal had reduced infarct volume, which is basically the tissue die after occlusion of blood supply. We use medial cerebral artery occlusion and reperfusion model of stroke, and compared to ad libitum, the infarct size was significantly reduced in young animals as well as in middle-aged animals. But that effect is diminished in old aged animal. But one thing I always say, Dr. Madsen, when I talk about this study, in that old age or old animal, we started intermittent fasting when they were already, old, 18 months old.
Yeah.
And but so if you go back and start intermittent fasting from day one, 3 months old, might say, and maintain that for next 21 months or 24 months and induce stroke, you may see different results. So we have shown that for the first time that intermittent fasting protects against stroke-induced brain damage.
Although,
as well.
Yeah. And we do know, Gary, that with aging, I'm getting old now. And anyway, when people get older, they're less able to tolerate a lot of different kinds of stress. Heat stress, right? With climate change, that's a good example that, a lot of the people that die from, really hot temperatures are elderly.
Yeah.
That's like it. But, pretty much, and in the stroke model, that's a stress on the brain. And apparently your data suggests that, at least the way the study was done, that the brain cells are not able, for reasons we don't know, to respond as well to the intermittent fasting.
In old animals, right? Yeah,
in old animals.
Yeah. And however, I have to say that there are some effect of intermittent fasting when you look into the protein and neurological deficit. There is still some effect, but not as much as you saw in young and middle-aged animals. So that was the first study. And then I moved to, I started my own lab in Texas Tech, and then moved to Australia, same university where I did my PhD. And I wanted to continue this study. So I applied for ethics, to test 24 hours intermittent fasting, or every other day intermittent fasting. And the ethics committee came back and they said, no, it is too cruel in Australia, and therefore we can only allow you to do 16 hours intermittent fasting. So my mind, I'm not sure if I'm wrong, but I always thought that was the first ever study to test 16 hours intermittent fasting in any animal model.
I think you're probably right. One comment on the So universities or National Institute of Health and so on, all animal studies, you have to put in a proposal, kind of detail exactly how you're going to handle the animals, if they're going to experience pain, how you're going to deal with that, and so on and so on. And yeah, even at the National Institute on Aging, believe it or not, our committee, when I first proposed to do the every other day food deprivation, they said that I need to justify doing that. And I turned it around on them and I said, well, this is known to increase lifespan and improve health. I think everybody else who's not doing intermittent fasting in their animals should justify why they're not doing it.
Yeah, that's right. that was a good one.
and it's and this is an important point though, that people should be aware of, that the normal conditions for lab animals is they're couch potatoes.
Yeah.
They're in small cages, they don't get much exercise, they overeat. I mean, they have food available 24/7.
They are practicing sedentary lifestyle basically. That's right. So they approved my ethics application with a 16 hours intermittent fasting. So I started testing the effect of 16 hours against ischemic stroke. So I basically repeated the experiment I did in your lab in 2004 and 5. And what we found, 16 hours also protect you against ischemic stroke induced brain damage, neurological deficit, and we also looked into this mechanism. And in fact, I worked with Alexis Stranahan at Augusta University, and we did some work in her lab, and we published that work in 2011. And so I wanted to continue, testing the effect of intermittent fasting against stroke, as well as study the mechanism, how intermittent fasting protects against stroke-induced brain damage. And this
and you should point out, in the study you did in my lab, you did look at a few kind of selected, we had kind of educated guesses on what kind of neuroprotective changes at the molecular level might occur. That's
right. And we looked into, when we gone to the literature, specifically your study from, I want to also mention that your first study from your lab testing the effect of diet restriction, in this case, color restriction, against stroke was, as you mentioned, published in 1999, And that's the only study before we started testing the effect of intermittent fasting. So then I gone through your papers and some other papers to understand, and also, of course, a lot of ideas came from you, Dr. Madsen, looking into the neurotrophic factors, heat shock proteins such as heat shock protein 78, GRP, and also HO1, and also cytokines, anti-inflammatory cytokines. So we approached our colleagues at main campus at NIH, and he had a really nice method with a little amount of lysate. He can study the expression of protein. So we obtained a lot of data that showed that intermittent fasting, increased level of neurotrophic factor BDNF and FGF, as well as its increased heat shock protein, and also reduces pro-inflammatory cytokine, increase anti-inflammatory cytokine. So we learned some insight from those tissue. And it's similar to what you have published with other model. I remember one of the work by Virendra, while I was there, looking into the effect of intermittent fasting against dementia, Alzheimer, I think triple transgenic mice, right?
Yeah.
And that was the first paper, if I can remember, in the field to show that intermittent fasting protects against dementia.
Yeah.
And I'm not sure you have gone through. There's one paper published in Nature Aging in 2022, basically showed the similar effect what you and Virendra published in 2000. When it was 2000. 7, I guess, right?
Yeah,
some
I
time.
think that's right.
Yeah. So with this study in your lab, we showed that neurotrophic factors increase, protein chaperones increase, antioxidant protein increase, and also anti-inflammatory cytokines in response to intermittent fasting. And we also show that intermittent fasting reduce pro-inflammatory cytokines. So we did look into that, at least not all of them, and some of them in response to 16 hours intermittent fasting. And we did see the similar effect as EOD, every other day intermittent fasting. But I moved to Singapore, as you mentioned. I moved around. I moved to Singapore. I still want to continue my work into this field. So, but I was thinking, Dr. Madsen, like, okay, we study about number of different proteins, and we know, but there's much more than that. Intermittent fasting, it's a pleiotropic mechanism. Probably, we are not seeing the whole picture. So how we can study that? That's where I started doing RNA sequencing from the tissue collected in response after intermittent fasting or after stroke. First study was using stroke as a model and studying the effect of intermittent fasting, looking at the gene expression. Yeah, one more thing I would like to also add, that when I give this talk about effect of intermittent fasting against stroke and dementia, the one question people ask, the time of intermittent fasting, and I'm pretty sure you came across this question as well. If you look into my papers, I fast them at nighttime. These are nocturnal, so. They ask what happened if you fast other way around, like in the daytime, and let them eat at nighttime. And there's a lot of work by Sachin Panda, is that right? Sachin Panda and some others about circadian alignment is important for the effect of intermittent fasting. So recently, it's not published yet. I would like to share with you. Raghu Bigumanti from University of Wisconsin, and I also part of this manuscript which we submitted recently, looking at the effect of intermittent fasting. So we had 3 groups ad libitum as usual, and the second group is active phase intermittent fasting. So intermittent fasting at nighttime, 16 hours intermittent fasting, and the third one is inactive phase of intermittent fasting. And the data was quite interesting. Both intermittent fasting had effect in many outcome, or disease outcome. But what we found, the effect of nighttime intermittent fasting, more significant compared to ad libitum, and also the inactive phase of intermittent fasting. So the circadian alignment is crucial for the induction of cerebral ischemic tolerance by intermittent fasting. It's quite interesting. And I think it's kind of support finding by others who are studying intermittent fasting and circadian alignment. So
okay, and then what are some? So you did transcriptomics. You looked at messenger RNAs for essentially all the genes, or nearly all the genes in the whole mouse genome. And what were some of your findings that you thought were particularly interesting?
So the question we ask, so I want to look into the whole picture. So the question asks, does intermittent fasting induce transcriptome changes that exert protective effect against brain injury? And if so, how are these effects altered under different intermittent fasting conditions? So we want to test 12 hours, 16 hours, and 24 hours. So first thing, I compared basically the normal ad libitum against intermittent fasting condition. The data was quite amazing. So we found a number of genes that basically influence cell-to-cell communication, neurogenesis, and neuroplasticity, and nervous system development, regulation of cell differentiation. I don't want to go through the gene name each. I'll tell you a few genes that I thought important. and also positive regulation of metabolic process. So when you get this big data, you can analyze using, a number of different data pipeline to learn about the signaling cascade that affected in response to intermittent fasting. So quite interesting. These pathways kind of, support the idea intermittent fasting is beneficial to the brain cells or neuronal cells. The second set of experiment what we have done.
One question.
Yeah.
When did you kill the animals in relation to was it the end of the food deprivation period?
Yeah, that's also a very important question, because there are some thoughts that the time of killing these animals, sacrificing these animals is important. And more recently, last month we collected tissue to answer that question, if there's any difference if you kill. But at the time when we collected, immediately after in the morning, immediately after intermittent fasting and ad libitum, we did not put them under, like overnight fasting to make sure that some of the genes regulated by just, immediate fasting. So that is important question. But over the years, we also collected tissue intermittent fast animal after they finish eating at night time, just to see. But the major set of pathways and genes what we see, these are in response to chronic time-restricted feeding or intermittent fasting, not immediate fasting, just overnight fasting or something like that. And also we tested the effect of a few days fasting, a couple of weeks fasting, and 1 month fasting, and 1, 2, 3 months fasting against ischemic stroke. And the effect, you can see after 1 month fasting, there's a protective effect. And in fact, Raghu also repeated this experiment at the University of Wisconsin. 1 month intermittent fasting has beneficial effect against ischemic stroke. So,
but if you go, so there's 2 week to 1 month period in terms of seeing beneficial effects on the brain, has held up in a lot of the studies, at least, from various people in my lab and others over time. Yeah. So I think this is interesting. Yang Lu, did you ever meet Yang Lu?
I think he left when I started.
He was an electrophysiologist, but what he did is he put mice on every other day food fasting, every other day fasting for 1 week, 2 weeks, a month, 2 months. I think that was right. And then he had ad libitum fed control. And then he euthanized them, took out their hippocampus, the brain region we study a lot. It's important for learning and memory. It's a lot of degeneration in Alzheimer's, epileptic seizures. Yeah. But anyway, what he found was that not until at least 2 weeks, and then clearly by a month, he saw there was kind of an enhancement of what we call GABAergic tone. So enhanced inhibitory control of neural networks, which kind of makes sense because it protected against epilepsy, which is out of control activity. And so whatever molecular changes are occurring at the gene expression level, or maybe protein or epigenomic, maybe we can talk about that in a little bit, it takes a while. And that kind of argues against that, like there's really, if you just fast for 1 day, all of a sudden your brain's going to be protected against a stroke. We don't see that. No. And so whatever changes occur, probably they involve not only changes in gene expression, the proteins encoded by those genes, but also even changes in structure. Yeah. You mentioned Alexis Stranahan, right? She saw effects on increasing number of synapses. Yeah. Or enhancing neurogenesis. Yeah.
And the work with Alexis Stranahan also not published, and we submitted to one of the high impact journal, and they rejected. Alexis Stranahan wanted to answer the question. It was there for like 5-6 years. Hopefully we'll get that out one day. And where just want to add that then we basically looked into the spine density, and we do see, at least in response to stroke, there's a significant reduction in spine density in the hippocampus, other side of the brain, contralateral. So if you induce stroke ipsilateral, we looked into the contralateral just to make sure that, because the ad libitum mice had larger stroke that may directly affect the hippocampus. Therefore, we went the other side to make sure that there's no bias, right? And what we found, there's a reduction in ad libitum, significant reduction in spine density, but that was not the case in intermittent fasted animals. So it was able to maintain the spine density. And that's quite interesting. I would like to also say a little bit about, after talking about the transcriptome data, what we have done with the electrophysiology work at NUS, National University of Singapore. Quite interesting. And we looked into the hippocampus of intermittent fasted animals. So coming back to our work studying transcriptomic changes or differentially expressed genes. So we did stroke in ad libitum and intermittent fasted for 16 hours for a few months, I think around 4 months. So our experiment mainly 4 to 6 month intermittent fasting. And you see amazing changes. So immediately after ischemia, 1 hour ischemia, in ad libitum, we saw around 40 genes were differentially upregulated in ad libitum. And when you looked into the brain of the tissue, when you analyze brain tissue of intermittent fasted animals, we saw 2,000 genes. So it kind of stress you are giving, they're able to respond significantly. And then you looked into these genes, and these genes are basically, anti-apoptotic, the genes that promote neural communication, neurogenesis, and also anti-inflammatory. So it's kind of
mitochondrial function or stress resistance.
Yeah, it is amazing to see that these intermittent fasted brain is already primed for stress condition like this too, and I think that's where the protection coming from. So the protein expression, we did not do proteomic those days, but we are doing now. We have access to the proteome next door, proteomics facility next door. So we are looking into the changes in the brain and other tissues. So that was
daily time restricted eating, so every day they're without food for 16 hours. So they have an 8-hour eating window each day. One thing that, we've hypothesized, based on our work and others, that the reason both exercise and intermittent fasting, and also, I guess, intermittent and intellectual challenges like you and I are doing now, are beneficial is during the challenge period, during the exercise, during the fasting, during, keeping your neurons real active, trying to figure out, you're trying to figure out what I'm saying, and then think about what you're going to say. But then the cells need a recovery period. Right. So everybody knows this for exercise, right? And your muscle cells don't get bigger during the exercise. They get bigger when you rest and eat.
Of course.
And sleep is very important too. And we think the same is true with intermittent fasting. During the fasting period, cells kind of adopt a stress resistance, conserve resources sort of situation. So maybe autophagy is going up, and to prevent buildup of toxic proteins like in Parkinson's or Huntington's. And then during the recovery period, we have some indirect evidence anyway that the number of mitochondria in individual neurons actually increases. So with intermittent fasting, over time, you get an increased number of mitochondria in neurons. and associated with that is increased number of synapses.
Yeah.
So it's kind of like your muscle cells. Again, they repeated cycles of the stress of the exercise and recovery, stress recovery. Your muscle, you can't just go out one day and exercise and all of a sudden your endurance has increased notably, or your muscle strength. It takes repeated cycles over time.
Yeah.
And that's what we're seeing with intermittent fasting too. But the recovery period is very important.
And I agree with you when you talk about intermittent metabolic switching. I just want to make sure for the audience, is that the term you introduced, intermittent metabolic switching, or it was in response to?
No, I don't think so. I think that was introduced by physiologists and people who study energy metabolism.
Okay,
but it's probably the first time with respect to the brain, maybe.
Yes, that's right. Yeah, I was looking, to the literature who introduced this term.
Well, that's a good thing you brought that up, because neither you or I have mentioned the word ketone yet.
Yeah, that's right. That's where I'm coming now.
And that's kind of the definition of fasting. If ketones go up in the blood, that indicates this metabolic switch from liver-derived glucose to fat-derived ketones is happening. Yes. So that's what the metabolic switch is. And we can talk about the ketones. They're a really good energy source for neurons, but there's also evidence that they actually affect gene expression, at least of some genes. I had one postdoc, Christina Morosi, who found in cultured neurons that beta-hydroxybutyrate ketone produced when you're fasting will stimulate BDNF gene transcription. Yeah.
I'm not surprised that Dr. Madsen, because this is basically environmental change to our cells, right? Like you have glucose to ketone, and so that's where epigenetics starts. So I was thinking about the mechanism.
Let me say one more thing that I haven't mentioned, and it's important.
Yes.
So everything we're talking about makes a lot of sense from an evolutionary perspective.
Yeah.
Because during evolution, animals whose brains and bodies function better than other animals in their, of the same species in the same environment, they had a survival advantage, right? If your brain and body don't function well when you haven't eaten for a long time, you're not going to be successful, and your genes aren't going to be passed on. So over, in the case of humans, a couple 100,000 years, but obviously this goes way back before humans.
Yeah, of course.
Animals evolved so that they function really well in a food-deprived state, because food's scarce out in the wild.
Yeah.
And so I think that's a really important point, that that's why there's so many positive effects of this kind of eating pattern, because evolution is geared our whole gene expression programs to kind of be optimized with that kind of a scenario.
Absolutely. It goes back to I'm pretty sure you came across Richard Dawkins' selfish gene. Well,
you can go back to Charles Darwin. Before they even had genes. But
and just one point I want to make. I was reading these days a lot of history books, and I mentioned about this one, and for the audience, like during the evolution, like 3.8 billion years of from single cell to multi-cell organisms and all the way to us, the environment played a major role in molecular evolution. When I say molecular evolution, about molecular genetics. So the epigenetic, basically, it's one of the oldest mechanism, that produce us today. And when you intermittent fast or calorie restriction or practice calorie restriction or exercise, these are environmental changes to our body cells, cells in our body. So they do prepare. And what was missing, how exactly intermittent fasting that lead to metabolic switching and ketone as an energy source driving that genetic changes, it's not known, right? Like so we thought, okay, we had access to RNA sequencing, and at the time, it's not expensive as doing chip sequencing or whole genome bisulfite sequencing. So we started with RNA sequencing to understand about the differentially expressed gene in response to intermittent fasting. And the next question we want to always ask, what's before that, right? And that's where our research now focusing on it. And so again, just to make sure that in response to 16 hours intermittent fasting, after stroke, what we have seen that intermittent fasted brain was more active when it comes to producing genes that involved in cell communication, cell differentiation, neurogenesis, and generation of neurons, and neuron differentiation, synaptic transmission, all these things. So quite interesting data. But as you say, Dr. Madsen, it's important to go after one or two genes to knock out and see that effect really coming from, the protective effect you see coming from that set of genes. So that future probably we will look into that those genes carefully. One of the gene I would like to mention, oxytocin. That's one of the the highly differentially expressed gene you see in response to intermittent fasting, and we all know about oxytocin, right?
Yeah, I had a podcast on that with oh my gosh, Larry down at Emory University. And he did a lot of pioneering work on the love hormone, or the love peptide, right? As social bonding thing. Yeah, there was a lot of interesting things. But so oxytocin was up with the daily time restricted eating.
Yes. That was so I looked at the top 10 genes, like that was on top of the gene list, oxytocin, in response 6 intermittent fasting.
Interesting.
Yeah, interesting. Yeah. So, but there are studies, right? Oxytocin is beneficial in stress condition or neurological disease condition. So that go along with the literature what we have out there. So one other thing I would like to also mention that intermittent fasting group exhibited extensive upregulation of genetic pathways involved in neuroplasticity. So that bring back to the question, and you just mentioned that one of your postdoc looked into the electrophysiological properties in response to intermittent fasting, right? And so we want to look into what this mean, neuroplasticity gene. So I gone back to Alexis Tranahan's data where we found spine density increase in response to intermittent fasting. But I also want to look at the electrophysiological properties. So I had the opportunity to work with Dr. Saji Kumar Sridharan. He was trained by Juliet Frey, one of the well-known electrophysiologist and neuroscientist in Germany at the time. And I think she moved to Augusta University for a while and came back to Europe now. And he was trained by Juliet Frey and Richard Morris. We all know Richard Morris, right?
Morris water maze.
Yeah, Morris water maze.
The classic test for spatial learning and memory.
Exactly. And so both of them, Richard Morris and Juliet Frey, introduced this the idea of synaptic tagging and capture, which basically, you remember the wine or the dinner you had on your wedding day, but you probably can't remember the dinner you had a few months back, because there's associate memory. There's two like different events happening at the time. One kind of
an emotional contact. You remember things, whether it's positive or negative emotions. You tend to
remember. Yeah. So there is so we wanted to study that scenario in response to intermittent fasting. So we looked into different type of intermittent fasting: 12 hours intermittent fasting, 16 hours intermittent fasting, and every other day intermittent fasting. So collected the hippocampal slides. And so the way you do this experiment, you have two electrodes in a different region in CA3. And CA3, for the audience, the part of the hippocampus, well studied. So we activate the first electrode that's not, it's relatively weaker stimuli compared to the second set of neurons. So that, of course, lead to EPSP, which is, electrical activity, and that maintains, so the curve go like this and maintain for a while, and then goes down to the basal level, right? Because the neurons start firing. And so you can measure it. And after 2 hours, you stimulate the second set of neurons. They are communicating with the first set of neurons, and quite with a strong stimuli, like 3 times stronger than the first set of neurons. And so what you see so the second set of neurons start firing, and you see again electrical activities of those neurons going up, and the EPSP quite high. And nothing happened to the first set of neurons. They go down all the way and reach the baseline by 5 hours. But in intermittent fasted brain tissue, hippocampal tissue, every other day, and I have 16, the first set of neurons, the firing EPSP, coming back after the second set of stimulation of second set of neurons. So that's quite interesting. And we did not see that in intermittent fasted 12 hours hippocampal slide, but did see that in EOD every other day, and I have 16 slices. So it was quite interesting. And then going back to the extended synaptic tagging and capture. So what we think that early long-term potentiation in the first set of neurons got transformed to late long-term potentiation. And so it's quite interesting.
Well, so maybe this is some electrophysiological correlate of the transfer from short-term memory to longer-term memory.
I think it's coming back to the gene expression and the protein that set up the neurons have in response to intermittent fasting, right? That helping them to bring this the memory back home. And we thought, what are the mechanisms? Maybe they related to amount of protein there, or protein being produced during that time period when we do the experiment, or BDNF, for example, so we blocked BDNF as well as protein production synthesis during that time frame, and then we saw that going back in the intermittent fasted.
So you used an antibody to BDNF or what?
Yeah, antibody to BDNF. Yeah.
Huh.
And so it was quite interesting. We used a mid-teen for blocking the protein production or synthesis, and TRKB antibodies, so FC.
Oh, okay. So antibody against the receptor to BDNF.
BDNF, yeah.
Yeah.
So you see that it's going back as you see in ad libitum. So, but I think that there is more than what we have seen in this experiment. Like, so we need to go back. So I want to do this experiment not only in intermittent fasted animal, also in their offspring. So for a long time, I want to come back and also study. There are, technology is quite amazing these days. So we want to collect the tissue and look at the genes at different sites, and there's a terminology for that, spatial transcriptome, or what's that called, Dr. Madsen? Something. Yeah. So you can
transgenerational effects.
Yeah, I know that's what I want to do, but to study the expression of genes in individual cells. So single cells, but in the slides, I'm sorry, I can't remember that. So you can collect the slides and do the expression of genes in slides. And
it's single cell transcriptomics.
Single cell transcriptomics, one thing. I think I'll come back to that.
Okay.
But
to back up a little bit, so epigenetics. So this goes back a lot to a guy named Lamarck.
Lamarck, yeah, of course,
several hundred years ago.
Yes,
a French that he wrote this idea that essentially the behavior of the parents, or some environmental experience of the parents. It could be something negative, like traumatic stress, or it could be a positive thing like regular exercise, or engaging in intellectual challenges. So the idea is that what the parents do can affect the offspring's gene expression. That is, the amount the gene is turned on or off. So without any change in the nucleotide sequence of the DNA, there can still be effects across generations in the amount of mRNA and protein produced from any particular gene. And this has been shown in animals so I mentioned like chronic stress paradigms.
Yeah.
So the idea is that, okay, obviously, so a baby comes from an egg and a sperm, right?
Yeah.
The DNA the woman's DNA in the egg, the man's DNA genetic material in the sperm, fertilized. So these epigenetic changes, which are kind of molecular modifications of the DNA that affect gene expression, for this to be true, this epigenetic mechanisms, then there must be some change in either the egg DNA or sperm DNA, or maybe in some early stages of embryo development even. Yeah, but anyway, this has been shown. For example, if you subject a female mouse to really bad types of stress, when before and during pregnancy, and then when the pups are born and you let them mature and stuff, they have more anxiety-like behaviors.
Yeah.
And
I mean, that's also known in human Dutch hunger winter study, right? And and they looked into the children of these mothers.
But the thing is, Gary, so in the animals, you can do what's called cross fostering. Well, you can do it in humans too. It's called adoption. But so in the animals, you can rule out any early life behavioral effects by, or you can even now with the modern methods, you could even take the early embryo from the female that's been stressed and put it into the female that hasn't been. But you can do these really important controls and show that this doesn't have anything to do with how the parent is interacting. It doesn't have anything to do with the early life environment of the offspring. It's something transmitted. Yeah.
There's one study published in Nature Genetics in 2016 showed that the high-fat, father and mother, and they got the sperm and egg and put that in the foster mother, remember that study, and it showed that the sperm and egg came from high-fat mothers or fathers had epigenetic effect.
The pups, their pups are more likely to develop obesity.
And
also it was a diet. So you can feed rats or mice kind of like a McDonald's diet. High saturated fat. You can even put fructose in their drinking water or glucose, and they will become obese and get diabetes. And that's not good for the offspring. It's not good for their brain either, because there's a lot of evidence emerging that, at least for women, if they have obesity and or diabetes during pregnancy, their offspring are more likely to develop an autism, autistic behaviors type scenario.
And these are, as you mentioned, known. The mechanism is not well studied, right? So, and also we don't know intermittent fasting helps or have phenotypic effect in offspring. So that's the question now we are answering while we are talking the experiments being done here in my lab. Now I'll share with you some of the early data what we got, and it's just like, I always quite cautious with this data because we are repeating now the cohort 2, to make sure that what we see from the cohort 1 is correct. And I will confirm that data soon, within the next couple of months. Of course, I will share with you immediately once I get this data. But yeah, but going back to our neurological, disease condition, stroke, and looking into the transcriptome. And meantime, we were asking this question, Dr. Madsen, we also want to understand more about other organs such as heart, liver, and muscles. And we started doing transcriptomic studies using these tissues. And as you know, that we have just published a paper in eLife where we are looking into the transcriptomic and proteomic changes of the heart, right? So it's not like organ specific. It's basically helping. I mean, if you look at the data, I could say helping all organs of our body. And when it comes to heart, we found one of the pathways, cyclic GMP PKG signaling pathway, right? And that is in response to 12 hours intermittent fasting, 16 hours intermittent fasting, and 24 intermittent fasting. So it's quite common to all types of intermittent fasting. But we also see unique and common pathways and genes of these different types of intermittent fasting, not only in the brain, but heart, muscles, and liver. So intermittent fasting time period is important. And I'm not sure which is the best, but coming back to intermittent metabolic switching, it's important to make sure that, we start producing ketone as an energy source from liver fat, right?
Yeah.
Or fat. And that change is important to drive these, the molecular pathways, what we see. And that's what my common understanding based on what we have done so far.
You mentioned Ruchin Wan, who was a senior scientist in the laboratory at NIA, and he had done some work in collaboration with Ed Licata, who's the chief of the cardiovascular group at NIA. And Ruchin found that the every other day food deprivation, when the animals were adapted to that, their resting heart rate was reduced and their blood pressure was reduced. And then he have these really neat setups where you can implant electrodes, one in the heart and one in the descending aorta, I think, for the blood pressure. And you can monitor heart rate and blood pressure 24/7.
Yeah.
For as long as you want.
Yeah.
So he had tons of data, and so he'd get baseline data, then put them on intermittent fasting for a month.
Yeah.
And heart rate and blood pressure down, not right away. Again, it took like a couple weeks, and then it stayed down. And then, but then when he switched them back to ad libitum feeding, no longer on intermittent fasting, then within a few weeks, heart rate and blood pressure went back up. But this cyclic GMP, the reason I was interested in that is it's actually the way that nitroglycerin relieves cardiac pain in people with atherosclerosis or atherosclerosis. It relaxes the blood vessel. Yeah. And it can lower blood pressure. If you don't get the dose right, you can actually faint. Yeah. But nitroglycerin, it's called a nitric oxide donor. Anyway, it's essentially you get nitric oxide produced, and it will increase cyclic GMP levels, and that's what relaxes the blood vessels. So this effect, the effect you saw in the heart, and your pathway analysis, may explain some of the effects we'd previously seen on blood pressure anyway.
Absolutely. I also came across clinical trial targeting this pathway in the heart failure condition. So I think it's already the community in this area.
Yeah, the Viagra, the sildenafil, right? The Viagra, was initially approved for erectile dysfunction, and because it relaxes the blood vessels in your penis so they get more blood. But you're right. These drugs are, it's actually also used for this drug for pulmonary hypertension. Pulmonary hypertension, it's approved.
Yeah.
But there are many studies, including looking at effects on cognition. Don Ingram, right before he retired, he did one study in rats and had evidence that this Viagra, sildenafil, would improve learning and memory in older animals. And he published that. So they seem to be safe. And I don't know, maybe they're mimicking in part the effects of intermittent fasting, according to your findings.
Absolutely. We need to do a little more mechanistic study to understand, I guess. So after we started doing, transcriptomic studies and understanding a little more about the mechanisms of intermittent fasting, and we wanted to look into, what regulates gene expression. And so we asked the question, does intermittent fasting induce epigenetic changes that subsequently lead to changes in gene expression? So that question I thought is important, and I looked at the literature. There's no study other than Linda Partridge and a couple of the paper published what happened. Oh, your paper with the Zhang Ru, that was looking in the gene expression, not epigenetic, right?
Right.
That was gene expression. But I think Linda Partridge paper looking into the epigenetic changes in response to calorie restriction.
But that was in worms or flies or something. It wasn't in
not in mice. I need to, it's a current biology paper. I can't remember exactly, Dr. Madsen, but there's not much out there when I start, look. So we want to look into epigenetic mechanism, but it's a question where to start. It's such a complicated epigenetic. When you talk about epigenetic, you can either look into DNA methylation or histone modification or microRNA, long non-coding RNA, etc. Right? But recently, I went to a GRC, Gordon Research Conference, Epigenetic, on the way I came and met you, right? And so it is quite a lot, known about epigenetic mechanisms and possibility of these mechanisms being inherited. And also evidence is in mammals, not very solid. We still need to learn the mechanisms of inheritance. And quite known in the field of epigenetic how gene regulation or gene expression regulated by epigenetic changes. That's we all know, right? So we want to start somewhere, and we start with the histone modification. We want to and start again in the brain and after stroke or before stroke, and comparing ad libitum versus intermittent fasting, then what happened in response to stroke or dementia. So the first study we published in neuroscience, right? In that study, what we looked into 16 hours fasting and every other day fasting. So after 3 months, we collected brain tissue and used H3K9ME3 antibody to study about the changes in that histone H3K ME3, H3K9ME3. And so it is quite interesting. We do see such a significant epigenetic changes. And then we also want to look into what happens if you bring these mice back into ad libitum diet, right? So the changes you see maintain even if you go back to ad libitum. So we asked that question as well. And so we do see epigenetic changes at 3 months after intermittent fasting. And we also see some of these changes are maintained even after 2 months of ad libitum. So mainly the clock genes and metabolic genes. So we did the integrative study as well. We also did the transcriptome analysis from this same tissue and look into, is that also true when it comes to transcriptome? So you do see there are changes after intermittent fasting, and some of these changes are maintained if you go back to ad libitum. And we don't know in human. I mean, it's quite interesting when you said that routine study, when you put them back in ad libitum, right? It disappearing after a couple of weeks or so, right? Into the heart. But here we see mainly genes associated with metabolism, and also some of the clock genes are maintained. So we need to further study in order to understand how long these are maintained and if there's any phenotypic effect from these differentially expressed genes after going back to ad libitum diet. I mean, I thought it's quite interesting for the public out there. You know, if you practice intermittent fasting for a while, and even if you go back, don't worry, you still have some beneficial effect from the, you know.
Well, with exercise, you know, so say you exercise a lot from between the ages of 20 and 30.
Yeah.
And then, I don't know, whatever. You have kids, you can't exercise, you know, whatever, and you don't. You've definitely benefited a lot from that 10 years.
Yeah.
Right?
Yeah.
But in short term, as far as your endurance and stuff, you know, if you get in shape, say you're a runner and you're in real good shape, run every day, whatever, you know, and then you have some accident or something, you can't run, say you can't run for a month.
Yeah.
You get out of shape. By the end of that month of not exercising, you're, you know, almost back to where you were. So, you know, I'm kind of comparing it, and that's it. But you've still benefited from having, in the long run, in terms of longevity and from that perspective.
We don't know, you know, at least we see differentially expressed genes are maintained. Some of them are maintained, but is that also leads to some beneficial effect? We don't know. We need to study, and that's what I also want to do, and probably.
But then are you going to do transgenerational stuff?
Yeah, I'll just talk about quickly. After this study, we also want to look into DNA methylation pattern in response to intermittent fasting. So my PhD student Sharmili Selvaraji, the previous study done by Gavin from my lab at NUS, and then Sharmili, who is at MIT now and doing her postdoc, she looked into the DNA methylation pattern in response to intermittent fasting, and what she found, the hypomethylation. We specifically look into promoter agent and CPGI.
Hypo or hyper?
Hypo.
Okay.
Yeah. And
so reduced methylation usually means there's increased gene expression.
In expression, that's kind of correlate with what we've seen with our transcriptomic data, right?
Yeah.
And also we looked into some of these differentially methylated regions and understand more about gene expression, right? But we have also done in the study vascular dementia. So what we induce vascular dementia, where we put the coils around the both common carotid artery and reduce the blood supply to the brain by 30% and leave them for, you know, a month or two, and that leads to white matter lesion and neuronal cell death, that typical the vascular dementia hallmark you see in human. And so
and then do they have impaired learning and memory when you test them?
Yes,
yeah,
they do. And in fact, we also looked into the effect of intermittent fasting in response to cognitive impairment. And so we do see, they're quite interesting when you induce vascular dementia or, you know, chronic cerebral hyperperfusion, you see hypomethylation. So this condition basically from, you know, day one, increasing gene expression, so reduced blood supply, at least we see differentially methylated regions, right? And hypomethylation. And on the other hand, intermittent fast animal, basically other way around, we see, you know, opposite to what we have seen in ad libitum animals. And of course, we did analyze the hypomethylated region and the hypomethylated regions, and to more understand more about these genes. And it's quite interesting study, Dr. Madsen. As usual, we need to go further and understand more about this individual the genes that in response to the vascular dementia as well as the intermittent fasting. And since you asked, we also did not Morris water maze, but Barnes maze test in these mice. And what we found that, chronic cerebral hyperperfusion or vascular dementia basically led to cognitive impairment. Intermittent fast animal did quite well compared to ad libitum. So there's a protection against cognitive impairment as well. So that I thought that's the key data we want to, you were excited to see that. And also we looked into the pathological hallmarks, white matter lesion and neuronal cell death, and intermittent fasting basically protected against changes in the brain as well.
Yeah.
So it was quite, we published that last year in Thromnostics, and then we want to continue these studies. Of course, I want to do a lot more in this area, but also the burning question, as you just mentioned a few minutes back, we want to ask that are epigenetic changes induced by intermittent fasting inherited, and do they influence phenotypes of offspring? And that's the burning question. And so we designed an experiment where we have ad libitum mother and ad libitum father, intermittent fasting mother and intermittent fasting father, and ad libitum mother and intermittent fasting father, and ad libitum father and intermittent fasting mother. So we had 4 different combination, around 4 months fasting, intermittent fasting, and we produce offspring from these mating groups and separated them into male and female, and then further separated them into normal diet and high fat, high glucose, high salt diet. So it's like, we call it Western diet. I'm not sure that's the right terminology to use. So we had 16 groups at the end, it's quite a big study.
But are you going to do stroke in any of those offspring?
That's the future. That's the future. Not only stroke, and I want to do vascular dementia, stroke, as well as I want to study the hippocampal slides and do electrophysiology and repeat some of the experiment we have published already with the Dr. Saji Kumar at NUS. And so I was lucky, the university bought me electrophysiology rig, right? I mentioned about this one to you, and I'm setting up here. So hopefully in the future we'll study the brain cells from offspring. But the data is quite interesting. We haven't published yet. We are repeating. And so first of all, we want to look into the glucose, ketone, triglyceride, cholesterol, and CRP, the marker for inflammation, right?
Yeah.
And what we found, quite amazing set of data. So we started measuring from, baseline, and a couple of weeks later, like 2 weeks, 1 month, 2 months, 3 months, 4 months, all the way. And you don't see much changes between these groups, ad libitum, offspring from ad libitum, father and mother, or offspring from intermittent fasting, father and mother, or other way, combination of either mother or father, right? You don't see much during like first couple of months, but from 3rd month, you can see when they become adult, you do see the difference, where we found that weight also, at least if you look into female offspring, comparing to offspring from ad libitum father and mother, the offspring from intermittent fasting father and mother are quite reduced weight, there's a change. And also we do see decrease in CRP, triglyceride, and so quite exciting data, but early days we need to repeat and make sure that, so we have now n equal to 20 to 30 from each group. So we'll have solid data, 32 parents. We looked in the parameters from 32 parents in each group, and we will have around 30 to 40 offspring from each group, from 16 different groups, right? And so I want to make sure that what we're seeing is true,
Yeah.
And so
that's exciting. Do you know of so then we come to the question of what about humans? And now there were some studies, the stroke studies you did in my lab, and a lot of yours, you do the intermittent fasting before you do the stroke.
Yeah.
But then the question comes, what about someone or some animal that's already had a stroke? Would they have any benefit of the intermittent fasting if it's initiated after the stroke? And there's at least a couple studies out there suggesting maybe, yes, they may have better recovery. And then, were you in my lab when Tang was there? Yeah.
Yeah. I was the mentor for Tang, right? You know?
Yeah. Okay. So I don't know what happened, but he'd written up a proposal. He's in Taiwan. He'd written up a proposal. He's a neurologist in Taiwan.
Yeah.
I know he'd written up a proposal to do a clinical trial of intermittent fasting in stroke patients. I'm not sure what happened. I went on clinicaltrials.gov.
Yeah.
I can't remember, a month ago or something, and I think there was a trial from Korea, but there's at least
there are two trials.
One for sure, maybe two trials.
Yes.
So that will be interesting. And then as far as trials in, patients with mild cognitive impairment, Alzheimer's, Parkinson's, I don't know. It's interesting. This GLP-1 receptor agonists that get a lot of attention because people with obesity are taking them like candy.
Yeah.
But because they suppress appetite. But I had Tom Foltini in England, who did 2 clinical trials of these GLP-1 receptor activator in Parkinson's patients, and with positive results. So now he's got a big multi-center, I think, phase 3 trial going on. Alzheimer's, we have a study that Dimitrios Kapagiannis, a neurologist at NIA, did, and he has some interesting results with intermittent fasting in people at risk for Alzheimer's. And so we'll see about, translating what's
this
in
data
animals
to
to
human.
humans.
But it's quite positive at the moment. And there are 3 papers I always mentioned published in Cell Metabolism. All these are research papers. One of them testing the effect of 8 a.m. to 2 p.m. eating period, and then 18 hours of fasting. And these are pre-diabetes men, I guess. It had very positive effect and brought back to the normal almost, all the parameters. The second study looking into, again, I think 18 hours, I can't remember exactly how many hours of intermittent fasting. And that basically looking into the white fat to the brown fat changes, and that linked to the microbiome changes in response to intermittent fasting in human. A third study is every other day intermittent fasting. Quite interesting, Dr. Madsen. Its effect is quite significant in young. These are healthy men, healthy people, and that study basically also showed that it's promote heart, health. So there are 3 studies, and there are a lot more studies at the moment. I think that in the future we'll learn more about all these clinical trial ongoing. But the key question, as you just mentioned, we need to see the effect of intermittent fasting in human disease condition, right? That's quite some time.
There are some studies, Ellen Mowry at Johns Hopkins, she focuses on multiple sclerosis, and she's done a couple of preliminary studies that suggest potential benefit, and she's doing a bigger study.
Nice.
Anything with any disorder with inflammation, in theory. Another area is inflammatory bowel disorders. That, maybe going back to some of your first work on the gut.
You look into absolutely. And so we do have quite a number of studies in animals. It's important to see what happened in human, and unfortunately my lab doesn't have that facility to do human studies. But I'm more into in the future looking into the epigenetic mechanisms in response to intermittent fasting and the phenotypic changes you see in offspring. And
Yeah.
then I want to go back and look into the effect of parental intermittent fasting when it comes to cognition of offspring and also different disease conditions. So this is what I want to do for next 10 to 15 years if I'm still healthy and getting funded. But more importantly, in the process, I want to learn about epigenetic inheritance mechanism. That's the missing piece out there. Like people know that as you go back to Lamarck and all these recent studies, we do see epigenetic inheritance when it comes to phenotypic changes, but we don't know what happened, the sperm and egg changes, epigenetic changes in sperm and egg, and through the embryogenesis, you don't see these marks basically disappear, and somehow you get these phenotypic changes and so this big question mark out there. So I thought we can use this model to learn,
Okay, Gary.
Thank you.
We're in different times. You're in the morning there. It's about 6:30 p.m. here and about 10:30 a.m. there.
10:30 a.m. Yeah, that's right.
Yeah. So anyway, I'm going to sign off, and
thank you so much.
Appreciate you taking the time for this. I enjoyed seeing you again and talking with you.
It's a pleasure. Thank you so much, Dr. Madsen. Thank you.
Bye.